Good evening, and welcome to the first event in our 2022 speaker series, Surprising Science, Borrowed Ideas Leading to Unimagined Consequences. I would like to acknowledge and pay respect to the traditional owners of the land on which I am this evening, the Ngunnawal people. It is upon their ancestral lands that the Australian Academy of Science is built. As we share our own knowledge, teaching, learning, and research practices, may we also pay respect to the knowledge embedded forever within the Aboriginal custodianship of this country. I am Professor Mahananda Dasgupta, Nanda for short. I am a fellow of the Academy and one of the conveners of this series. I cannot tell you how delighted I feel to see actual faces in front of us today, and we hope that we will be able to bring you the speakers in person as well in due course. We are working on it. Today, I'll be joined by my co-convener, Professor Drew Evans from the University of South Australia, who is joining us tonight from Adelaide. Welcome, Drew. And I'll now hand over to Drew to introduce himself and talk about the speakers. Drew.
Thanks, Nanda. And equally, I'm delighted to be convening this series with you and coming to you from the virtual world. Today, I'm joining from Ghana country over in South Australia, and I'd also like to pay my respects to elders past, present, and emerging. Thank you for all joining us for our first event in the series. Tonight, we're going to hear about carnivorous plants and some inspiration from those plants from Dr. Adam Cross and Professor Chiara Neto. Before we hear from them, there's just a few things that we'd like to run through with you. So firstly, as opposed to asking people to turn off their phones and switch them to silent, we'd encourage you to firstly switch them to silent, but if you'd like to engage with the conversation on Twitter, please do so by using the hashtag #SurprisingScience. Now, for those of us joining remotely online, you'll be able to submit questions by scanning the QR code that's going to be on screen shortly. For those of you live in the face-to-face at the Shine Dome, feel free to also scan the QR code on screen, or also there should be a QR code on your printed program. And I think over the last few years, we've had plenty of practice with scanning these codes. And the way tonight's event will run is that the questions will be held until after both of our speakers have presented. And so with that, I would now like to introduce tonight's first speaker, Dr. Adam Cross. So Adam is a research fellow at the School of Molecular and Life Sciences at Curtin University. He has pursued a love of carnivorous plants since the age of 6 and is an international world-renowned authority on their ecology and conservation. His passion for their preservation has led to involvement in the conservation assessment and listing of over 250 species for the International Union for the Conservation of Nature. To hear about the fascinating world of carnivorous plants, please join me in welcoming Adam.
Wonderful to be with everyone today from Noongar Butcher here in Western Australia. I am a huge plant nerd. I've been captivated by carnivorous plants since I was only 6 years old, and they are the perfect group to excite people about science, or in many cases, to inspire some brilliant science fiction. They flip the table of what we really traditionally expect from nature. They don't sit there passively and allow insects and other organisms to eat them. They've really come up with these strategies of survival that just captivate our interest and really just change the way that we can perceive the environment in which we live. Now, most organisms in the plant kingdom are autotrophic, which is that they obtain nutrition through photosynthesis and the root uptake of nutrients. You know, we grow plants in the vegetable garden, and then we consume them. There's a cycle of plant productivity that we're used to. Now, some plants have decided that this is not cool enough for them, and that they are going to become heterotrophic, which is that they augment their nutrition not only from photosynthesis and from soil uptake of nutrition, but they find nutrients through other sources. And this can be parasitism in the case of some orchids and other species, or in this instance, carnivory. And I'm not the only person to have been captivated by carnivorous plants over the years. Perhaps the most famous is Charles Darwin, who wrote equally famously that he cared more about Drosera, which is a single genus of carnivorous plants that he was studying in the late 1800s, than he did the origin of all the species in the world. Now, that might seem like a fairly interesting and outlandish claim, but having been fascinated by these species for my entire life, I can see exactly where he was coming from. So, what is a carnivorous plant? What makes a regular plant carnivorous? It has 5 essential traits, this syndrome, and it's a bit of a gradient. Some species exhibit all of these traits, some, as we'll talk about momentarily, only exhibit a few. Now, the first trait that needs to be possessed is that they need to have a mechanism of capturing or trapping prey. That seems relatively obvious. Usually they use attractive specialized leaf modifications or traps to achieve this. But they also need to have ways of killing the prey that they capture, then digesting that prey, and then crucially assimilating the nutrients from those killed and digested prey into their own tissues and using those nutrients for their growth and development. Now, why this is an interesting gradient is that there are a great many species around the world that have developed what we describe as protocarnivory or paracarnivory as a mechanism to protect them from predation or herbivory. They have things like adhesive mucilage, which stops insects from eating their flowering parts, preventing them from reproducing. But crucially, a lot of these species, many of which we come across in our everyday lives, like plumbago or even petunias and even potatoes. One famous group here in Australia is the Stylidium or trigger plants. But crucially, many of these species may capture and even kill insects, but they don't digest them. They don't get this nutritional reward, which is the fundamental keystone of carnivory. And on top of that, there are several iconic species that many people, if not everyone, will be aware of. You know, the trumpet pitchers, the North American Sarracenia, and the incredibly iconic Venus's flytrap, Dionea muscipula from the Carolinas in the eastern United States. But did you know that that's not all? Did you know that there were nearly 900 species of carnivorous plants that are currently known, described from almost every continent on the Earth? and that almost every single year there are more and more species being described. They are one of the most fertile groups for botanists examining diversity and evolution. And did you know that almost a third of all species on the planet are found here in Australia, often literally just outside our back door? In Perth here, we are very lucky where we can take a couple of minute drive and find 20 or 30 different species of carnivorous plants just in the remnant bushland within the city itself. Now, some of these are minuscule. They're species that you would never notice unless you were actively searching for them, like this species of pygmy Drosera, the rosettes of which the trapping leaves are barely 4 or 5 millimeters across, and essentially the only part that is visible for most of the growing season are the flowers, which can often be incredibly showy despite the diminutive stature of the actual species. And we see a complete gradient of size and shapes and colors and morphologies all the way up to the really, really large tropical pitcher plants of Borneo and other areas of Southeast Asia, the Nepenthes. Many of these species produce traps that are large enough in some cases to catch spider monkeys and other small mammals. Even birds have been found inside some of the traps. And they can hold up to 2 liters of water. This is a pretty significant structure that has been evolved for the single purposes of trapping insects or other animal prey. Interestingly as well, there are species that have transitioned from a terrestrial life to a purely aquatic existence. Aldrovanda vesiculosa, which is a species that I've spent a great amount of my life studying up to this point, is a fantastic example where this species is entirely aquatic, but it employs a unique snapping trap very, very similar to the closely related Venus's flytrap, except that it operates only underwater and yet with one of the fastest movements in the entire plant kingdom. This species and also other carnivores from the genus Utricularia employ trap movements in the order of milliseconds from open to being closed. And this allows them to catch incredibly small insects in very fast periods in the underwater environment, which offers a completely different set of challenges for capturing prey. Now, that is, as I have spent most of my life captivated, incredibly interesting. And there are these huge diversity in form and function and structure, but there are also amazing groups that many people, even in the science and study of carnivorous plants, are less aware of. You know, the surprising fact that there are, in fact, carnivorous bromeliads. South America has several species of carnivorous bromeliad, which, depending on which type of systematics we look at, may be one of the most basal and simplistic forms of carnivory, where just the inward rolling of leaves and the development of digestive enzymes at the leaf bases has facilitated the pooling of water and then the attraction and digestion of prey. But there are also species being discovered even in the last few years, like this Philcoxia from Brazil, which is considered to lasso or otherwise adhesively trap small nematodes on its leaves. And these leaves essentially just sit below a fine layer of white quartz sand, which allows through enough light for photosynthesis, but also allows these nematodes to essentially swim past the traps and be captured. And perhaps most surprising of all, there are species of carnivorous shrubs and trees, 2 species of Rorigula from South Africa. And what's really interesting about this group is not only that it is carnivorous and not only that it is grown to this sort of size and stature where anecdotally we see small birds being captured, but it's also developed a unique mutualism with a hemipteran bug from the genus Pamaridia, and it relies exclusively on these hemipteran bugs to actually digest the prey that it captures for it. Because while it produces extremely powerful resinous mucilage, which it uses to trap the insects, it actually lacks the digestive glands required to then break down their tissues and absorb the nutrients without the assistance of this other bug. And this has raised a lot of questions among the literature about the 5 key strategies required to be carnivorous. And in fact, whether species like this that rely on another party to fulfill part of that carnivorous nature are indeed carnivorous or protocarnivorous. But however we look at it, carnivory is an extremely effective strategy that plants have developed for nutrient acquisition and survival. It has evolved independently, we think, at least 11 times, if not more, over the last 85 million years, which makes carnivorous plants one of the most phylogenetically and morphologically and structurally diverse groups of plants on the planet. There are many different kinds of traps. We have adhesive traps, snapping traps, suction traps, even lobster pot traps, and the pitfall traps, which I mentioned earlier on with Nepenthes. And I'll go into a little more detail about the pitfall traps and why they're so incredible just shortly. But one of the most interesting things in regard to the ecology and biology of carnivorous plants is that despite one of the key reasons why we're fascinated by them being that they eat insects or other animals, we actually know very little about what it is that they eat. We don't know, for example, whether species that co-occur together in close proximity compete for prey and whether that influences their chances of survival. And we don't know whether some of these species actually eat the insects that they rely on for the pollination of their flowers and subsequent reproduction in an issue that is termed pollinator-prey conflict. Now, if we look at some habitats and some sites where carnivorous plants are found in abundance, for example, for many species, very well lit, sunny, open, and often quite wet environments, we can find 10 or 15 species of carnivorous plant co-occurring in very small areas. This is an example here in the Perth Hills, a photo taken by my PhD student, Thilo Kruger, and you can see that there's a diversity of different species occurring within, walking distance of each other. They are likely capturing similar assemblages of prey, or at least they're exposed to similar assemblages of prey. And yet they are co-occurring and have likely co-occurred here for a very long time. Now, it's interesting because when we actually examine the strategies that these species utilize to co-occur, we find that even species with the same approach to trapping insects exhibit really remarkable differentiation in their ecological and morphological specialization. And we suspect that this actually allows them to co-occur, and in fact may have been one of the most significant drivers of diversification and evolution in this group. It allows different species to co-occur in the same habitat, and where we have different species co-occurring and selectively finding different areas of that ecological niche that they want to diversify into over long periods of time, and then when we have subsequent isolation of those populations or groups, we can see speciation occurring. One of the ways that we have examined this in the past is by analyzing the prey spectra, which is essentially the diet of different species. And traditionally, this is achieved through really laborious study, hours and hours and hours of taking photographs in the field and then examining those photographs back in the laboratory. These images were taken again by my PhD student, Thilo Kruger, in the far north Kimberley, which at this stage, the field site that we worked at was accessible only by helicopter or by the mail plane for a few weeks of the year. And so just obtaining these photos to actually begin studying the diet of the species was very, very challenging, both from a fieldwork perspective and also from a study perspective. But we've come a little bit of a distance from just simply examining photographs in the last few years, and there are now quite amazing approaches that we can use to look at these different prey spectra that different species exhibit and the approaches and specializations that they adopt in order to target different groups through approaches such as DNA sequencing. The metabarcoding of composite samples, where we might have a single leaf covered in different prey, we can sequence the entire leaf to get an idea of the different organisms that were present in that sample. And as you can see on the right, two different species, very, very closely related Drosera sundews, the adhesive trapped group here, very diverse in Western Australia. We see very, very different assemblages of organisms being captured by these two different species. Now, we don't know in some instances whether that comes down to different species being different colors or releasing different volatile compounds that might attract different organisms, different insects. And crucially, we don't know in some instances which organisms pollinate these species. So there are still a great many different questions that need to be asked, but it does start allowing us to draw inference between the different strategies that are employed. And if we come back to the pitfall traps, particularly the genus Nepenthes, which is a very, very diverse group of carnivorous plants, as I mentioned, from the tropics, we can actually begin determining the extent to which different species rely on carnivory and the extent to which they invest in the carnivorous structures that they adopt. So for example, we can see, this is a study that we undertook recently, which is to be published in Annals of Botany, where we have different amounts of nitrogen and carbon and different signatures in the isotopic composition of those elements, which can tell us the degree to which those different species are actually relying on carnivory, or the degree to which those species are actually similar to just reference organisms, reference plants that are occurring with them. And what we see in many species is extraordinary morphological specialization towards particular functions. We can see on the left here a group of 6 different Nepenthes, which all co-occur within a relatively small region in Borneo. And the size, the shape, the color, the structures that these different species employ is quite remarkable. On the right, just as one example, you see Nepenthes lowii, which has actually evolved to be something of a toilet bowl. It actually secretes a substance from the lid of the pitcher, which attracts a small tree shrew from the region, who, once it's had a lovely feed, then deposits its package of nutrients into the pitcher for the pitcher to obtain its nutrition that it requires to survive. And this, from the studies that we've done so far, appears to be actually as effective a strategy, if not more effective a strategy, than just the specialization towards catching insects. So this is a group of organisms that may actually no longer be carnivorous. They may have transitioned into another form of ecology altogether, scativorous or some other descriptor. But the Nepenthes are an amazing group because we don't just see strategies for carnivory, we also see what we describe as biological mutualisms. Now, the attraction of tree shrews to defecate in the pitchers is one such example, but there are 2 others. Just, very, very interesting examples where Nepenthes hemsleyana, for example, another species from Borneo, has evolved almost perfect bioacoustic domes at the back of the pitcher, which a species of bat uses to echolocate in the forest, and in flight will turn itself backwards, land perfectly in the pitcher, and use it as a roost overnight. Now, the bat receives a safe place to roost for the evening, and the pitcher receives the proceeds of whatever the bat needs to do over the course of the evening into its pitcher fluids. Nepenthes bicalcarata, for example, another species, actually actively encourages ant colonies to colonize its tendrils. The ants receive a safe place for their colony to exist, and the plant receives a fantastic soldier force for protecting it from other pests and pathogens. Now, one of the most unique and incredible carnivorous plants is a species unique to Australia, the Albany pitcher plant. It's perhaps 55 million years old. It's virtually never moved, if at all, from its highly restricted distribution down near Albany on the west coast of southwest Western Australia. And despite the fact that it looks very similar to other pitcher plants, it's actually genetically, evolutionarily closer to roses and cabbages than to any other carnivorous plant, including the other pitcher plants. It's a wonderful example of convergent evolution. Now, I talked about biological mutualisms. The Albany pitcher plant, Cephalotus follicularis, is one of the most complex examples of biological mutualism of any carnivorous plant. It has mutualisms not only with a species of ant from the genus Hydrodynamic, which we suspect may also be associated with its pollination, and of which the plant captures quite a few, but it also has a mutualism with a species of stilt fly, Bodicis ambulans. The stilt fly actually morphologically mimics the ant, the Hydrodynamic, and in its larval stage requires, we suspect, the fluid in the bottom of the pitcher plant for its incubation and its larval development. So there's this wonderful 3-way or possibly 4- or 5-way biological mutualism between different organisms that have co-evolved together to occur in just a single small location. Sadly, this species has become increasingly threatened by habitat loss, climate change, and even poaching, the illegal collection of carnivorous plants from the wild, which is also a problem that's seriously affecting Nepenthes. But it's also an example of the wonderful way in which plants have evolved specific structures that assist them in their carnivory, particularly surfaces. Now, we can see from electron microscopy of the trapping structures of the Albany pitcher plant that there are wonderful ways by which this species has gone above and beyond to make sure that it gets the meal that it desires. There are nectar glands on the outside of the rim of the pitcher and of the keel, which not only attract insects, but then attract those prey directly to the mouth of the pitcher. It has a hooked peristome with a very, very slippery, finely dissected surface, which then allows for the retention of prey once they have fallen into the trap. They have incredible microscopic structures on the inside of that upper trapping surface that ensure that prey are unable to get any sort of purchase once they're inside and are directed to or fall into the fluid below. And they even have fake windows in the lid of the pitcher to fool flying insects into becoming disoriented and falling into the pitcher fluid, where they are then digested and the plant absorbs their nutrients. Now, those are incredible structures from a biological perspective, from an ecological perspective. But if we think about the way that carnivorous plants have captivated us, through science fiction and through other really sort of fantastic ways they've captured our imagination, there are other ways that they have and continue to inspire us.
Well, thank you, Adam. I think everybody will in the audience will agree that that was absolutely fascinating talk. I think, for one, just hearing about the significant amount of carnivorous plants here in Australia, we tend to talk about how people coming to Australia need to watch out for the wildlife. To be honest, I think people need to be a little more mindful when they go for a walk, a beautiful walk out in the rainforest. Certainly, it's opened my eyes to the amazing things that plants can do just to get a meal. That's pretty impressive. And so with that bit of inspiration, I would now like to introduce tonight's second speaker, Professor Chiara Neto. So Chiara is a professor of physical chemistry and an Australian Research Council Future Fellow, and leads the Nano Interfaces Lab in the School of Chemistry at the University of Sydney. Chiara and her group focus on the physical chemistry of interfaces, a multidisciplinary field spanning the traditional disciplines of chemistry, physics, material science, and bioengineering. To be surprised by how their work is connected with carnivorous plants, please join me in welcoming Chiara.
Good evening, everyone, and thank you, Drew, for the introduction. It's a great pleasure to be with you, even if from a distance. I have been extremely fascinated by everything that Dr. Adam Cross has just told us, and I continue to be, and there's so much more that I would like to know about these fantastic carnivorous plants. But tonight I'll focus on one single aspect that has inspired our work. So as a physical chemist, I work on surfaces, and I'm particularly interested in exploring how surface structure and surface chemistry can affect the microscopic properties of surfaces, in particular, how they interact with liquids. And inspiration often comes from natural surfaces that have had, as we've been told, millions of years to develop the most successful, the most efficient strategies to achieve their goals. So by looking at nature in our research, we can really shortcut our research process and jumps right into a strategy that's sure to work. So that's what I'll tell you about today is bioinspiration that tell us how to do nanotechnology. Most of our inspiration for today's talk comes from one carnivorous plant, the pitcher plant shown here. As Adam has just told us, its rim is highly structured with the structure that you can see on the right-hand side. What the function of that is, Adam has already alluded to, and hopefully today you'll find some surprising facts and how we can learn from this fantastic species. A lot of what I work on has the hope to benefit the energy crisis of our planet. So as you know, a lot of research is spent nowadays in trying to source energy in a more sustainable way, for example, by using renewable resources. A lot of what I do is really trying to obtain applications and materials that allow us to perform the same function, but with less energy needed. And one way to do that, as I said, is to use surfaces. So you're all familiar with the idea of slippery surfaces. You've probably all seen this sign. When somebody has spread water on a smooth floor, you know that it's very dangerous because it's easy to slip. And that's because a thin layer of liquid may intervene between your shoe and the floor, and you have no grip, you have no friction. So a lot of what I'll be telling you today is related to this very effect. So the pitcher plant has a structured rim that makes it very easy under humid conditions, when it's rained or when the atmosphere is humid, to trap a thin layer of water within the structure. So on the right, I'm showing this scanning electron micrograph of the pitcher rim. In my talk, the scale bar will be very important, so I'll point to you when it's relevant. Here, the distance between these hooks that form the structure of the pitcher is of the order of 25 micrometers. So to make a comparison, the width of human hair varies between 20 micrometers and 180. So this distance here is about the width of your hair. The presence of that thin layer of water that's trapped within this surface makes this surface extremely slippery. So the prey touches the slippery surface, cannot hold on, and slides all the way to the bottom of the pitcher where the digestive juices are. So what we've done in our research is take inspiration from these topographical features, morphological features, and try and replicate it with materials that are now suited to modern applications, so that it will be more long lasting, that will hopefully have a lifetime that's longer than that of the flower. And we call these structures nanowrinkles because they are produced by the same mechanism that leads actually to wrinkles in our face. They are spontaneously formed when a substrate underneath this top layer shrinks. And in terms of the features, they recall those of the pitcher plant. The scale bar here is smaller. You can see 1 micrometer, but the principle that we're using is very similar. So the idea of this spine-inspired surface is we have a very fine structure. We infuse it now no longer with water, because most of the liquids that we will want to be slippery to will be water-based. So we infuse it with a lubricant that's mostly an organic synthetic oil, such as silicone oil. And now this combination produces a liquid interface that is entirely slippery to pretty much any liquid you place on it. The combination of the structure and the infused liquid is what we call the lubricant infused surfaces. So occasionally you hear me talk about LIS. So to give you an idea of what this looks like in an experiment, most of the surfaces around you, even if they're smooth and made of plastic, will be actually quite sticky to liquids. So if you put a droplet of water on the surface and you incline it, like if, you know, on the your windscreen, the water droplets will remain attached to it. So if we now drip some dyed water droplet on it, you can see that the water droplets stick to the surface, and you have to either shake the surface or somehow remove the droplets to get them to fall off. On the other hand, here on the right-hand side, we've done the same experiments with dyed water, but now on a slippery surface. And now the droplets roll off almost in a spherical geometry, leaving nothing behind. And this is a signature of what we call a slippery surface. So only a very small tilt angle is required for the droplets to roll off. Now have a think, what would you do if you had at hand a really good slippery surface? You might think of good pranks that you could be playing on people you don't like, but also very useful things that would have practical applications. So in the literature, there are fantastic examples of showing that these surfaces are self-cleaning. They're completely anti-adhesive to pretty much anything you want to put on them. So for example, they're anti-icing. They can prevent the formation of ice on the wings of airplanes, which makes them safer. We've shown that these surfaces repel the formation of blood clots and therefore reduce the occurrence of thrombosis on artificial surfaces exposed to the blood. Today, I'll be showing you examples of two other applications, repelling marine fouling and reducing hydrodynamic drag. Firstly, let me tell you a little bit more what these surfaces look like. This is a scanning electron micrograph taken over many tens of micrometers. You can see that we can prepare this surface over large areas, and it's fairly uniform, but it's not ordered. And if you look at the close-up here on top right, the width of the wrinkles is of the order of 200 nanometers, and their height is of the order of 200 nanometers. And then there's a larger scale roughness that's more around the micron. So 1 nanometer is 1 billionth of a meter, so 1,000 times smaller than the micrometer we introduced before. And this is really important. Size is really important, because what we want is really to have very small pockets, these areas that I'm highlighting with my mouse now, in between the wrinkles, that allow us to trap small reservoirs of lubricant all across the surface. So when we infuse this surface with a silicone oil, which is entirely non-toxic to cells and to marine organisms, we achieve a slippery surface. So this is a cartoon picture that I've shown you. Just to clarify, why do we choose this particular combination of material? The substrate is made of Teflon because, as you know, Teflon is non-stick. You probably all have a non-stick pan in your kitchen that's been selected because it doesn't interact either with water or with oils very strongly. Silicone oil is also has low interaction with most other chemicals, and it's fully spreads on the Teflon when immersed in water. So it creates the nice lubricating layer that we need to have this performance. The microstructure does the rest in retaining the oil. But in order to retain this performance that we really want this layer to be uniform and last for a long time. And of course, there's many situations in which this layer might be depleted and we might lose the properties we want. So a lot of my research over the last few years has been around how do we retain this lubricant long term, and how do we study how much there is locally, and basically how we get better materials this way. And for this, I'd like to give you a little physical chemistry lecture. Going back to the idea of capillarity. You're all familiar with the idea that if you have a glass capillary and you immerse it in a water reservoir, water will spontaneously be taken up the capillary, and also that the thinner the capillary is, the further up the water will travel. This effect depends directly on the curvature of this water-air interface. So if I describe that curvature with a circle, I can draw a radius that characterizes that circle. The smaller the radius of curvature of this meniscus, the higher the height that the water will travel. So the higher the pressure that pulls the liquid out. So in our surfaces, effectively what we have is tiny capillaries that are contained within the wrinkles that have tiny radius of curvature. And this is how you can visualize it. We take a lot of images using atomic force microscopy, as shown here. Here, the scale bar is 2 microns, and the height of the features is about the same. If we take a cross section on the surface, I'm showing it here with this gray substrate. We then infuse it with the oil that's shown here in yellow, and we can image the shape of this interface at the oil-water interface. And if I'm showing you here some data from these images, the radius of curvature that we can extrapolate is of the order of a few microns. And that corresponds to a very high retaining pressure, capillary pressure within these wrinkles, which explains why they're so effective at retaining the lubricant, even when exposed to flow. The technique that I've just shown you was developed in my lab and allows us to, as I showed, image at the same time both the wrinkle topography and where the lubricant is distributed. So again, if I show a cross section across there, I'm showing both, these gaps in between the wrinkles and where the lubricant is filling them up. And so crucially, as long as we have some liquid spread everywhere on these structures, our surfaces will remain slippery. It's really important that we can make these contour maps so we know how much lubricant is left at any one time, even when we've aged the surfaces or we've exposed them to some very harsh treatment. So we can draw a three-dimensional map of where our lubricant is and check that the surface is still working. So the first problem that we've applied it to is marine fouling. This is the accumulation of marine organisms such as algae and barnacles to the bottom of hulls or any immersed structures, and has huge environmental and economic consequences. For one, the increase in fuel costs in driving a ship through water when it's fouled are huge, of the order of billions of dollars a year. So if you can coat the ship hull with a slippery coating, then you can address this in a way that's environmentally friendly. So the first thing that we did is we proved that our surfaces repel the first stage of the fouling contamination, which is the attachment of bacteria. In this micrograph, I'm showing in violet the attachment of biofilms that occurs very readily on dry Teflon, but hardly at all on the infused surface. And even after the infused surface has been spun in seawater for 2 weeks, it retains its function. And importantly, we can achieve this type of behavior with a tiny amount of lubricant. Only about 0.2 milliliters per square meter of surface are necessary to achieve this behavior. So really effective retention and use of this lubricant within these surfaces. The second thing we did is we exposed these surfaces to a real environment. So my student Sam went to Watson's Bay, which is not a bad place to do research if you can do it. And he installed some surfaces under the waterline on some shark nets and checked the status of the surfaces again after 7 weeks. And the comparison this shows here in some images. On the left-hand side, we have a plain perspex surface, which gets fouled very readily and it's completely covered in algae. Further to the right, there's Teflon, which gets fouled, and about 60% of this surface is now covered in fouling after 7 weeks. And all the way to the right, I'm showing our best infused surfaces, of which only 20% is fouled, and then more than half of that can be removed when we simply wash the surface in water. So these surfaces are effective not only at repelling fouling, but they also foul release. So anything that attaches is fairly loosely attached and can be released. So the stage of this research now is that we're partnering with a company based in Sydney to try and fabricate this type of structures over large surfaces that could be indeed applied on ship hulls and similar systems. And the second application I'd like to talk to you about is the reduction of hydrodynamic drag. So if you imagine that these lines here are the walls of a pipe, and you have a liquid flowing from left to right inside, the velocity of the liquid will be maximum in the center, and let's say it would be whatever determined by the pressure that you're applying. But then some of that pressure would be, let's say, wasted because the liquid at the wall of the pipes is actually stationary. We say there's no slip. The liquid is stationary, and therefore we have to apply effectively more pressure to get this liquid to flow. And this effect is due to the fact that there is a finite friction between the fluid and the solid at the interface. Now, if we can include on this surface a coating such as ours that induces slip, means allows the liquid to flow freely at the wall, we now reduce the hydrodynamic drag substantially. The smaller the diameter of this channel, of this pipe is, the bigger the effect of the walls will be. And so this is particularly important on a micro scale, so microfluidic devices, for example, that are used in biomedical devices. So the aim is really to reduce hydrodynamic drag, to reduce the energy required for flow by using slippery surfaces. And the cartoon that is drawn here is actually taken from a real profile of the surface. So just to remind you, we have this structured surface, the space within the surface is occupied by a lubricant, and effectively we replaced a solid here in gray with a liquid here in yellow. And so the liquid-liquid interface has much less friction than you would expect for a solid liquid. So this was the initial premise of our work. And actually the consequences, or rather the results that we found, were quite surprising to us, because this picture was not confirmed. It's much more complicated than that. So I'll tell you a little bit about it. This is the device that we used mostly to do the measurements. We have a few that we've designed, a microfluidic device where you can see 5 channels made in here. We control the inlet and outlet of liquid, and we measure the pressure required to induce the flow at a certain rate. And we can replace the surface at the bottom of the cell with whatever surface we want to test, in particular liquid infused surface, relative to a smooth, sticky surface. And we found that liquid infused surfaces, in fact, reduce drag by up to 30%. So a huge amount, really pleasing result. And this effect is really long lasting. So as you can test this for 24 hours within this cell, and there will be no change as long as the rate doesn't change. At the same time, we conducted some computational fluid dynamic simulations to try and mimic the same geometry. And to our surprise, we found that the effect that you would expect if you just had the lubricant in place, so just the plain surface as we understood it up to that point, would never justify this huge drag reduction. And so this took quite a few more years of investigation to actually establish what was going on. And the paper shown here actually just came out in January. Very proud to show it, because it was the work of mostly Chris Vica-Sanchez, who almost has completed his PhD. We were able to show that, in fact, what happens on these surfaces is that bubbles nucleate. So here I'll show a brief video where the surface is upside down, the flow is underneath, and you see the appearance of these semispherical bubbles that grow with time. And these particular bubbles are very large because we are using highly gas water. You can see their profile and their top view here. So, you know, they're not nano-sized, they're micro-sized, but we can pick them up with confocal microscopy, as shown here. And what basically that means is we don't just have 2 phases as we were assuming to start with. We have a non-frictional interface at all, you know, no completely slippery, which is the gas water interface. And that's what explains the huge drag reduction that these surfaces give. Very pleasingly, we were able to do the same imaging with atomic force microscopy, which revealed that if you have even degassed water, so very little gas still present in the water, you still get bubbles. This yellow corner down here represent the air thickness that we were able to image in situ. And the thickness of this bubble is of the order of 100 nanometers. It's a few microns wide, so not easy to detect with any other means unless you have a nanoscale technique such as this. So the presence of the interfacial gas allows us to basically reduce the energy required for flow, and therefore brings huge energy savings. So I hope I've convinced you that there's a lot of interesting surprises to find when you're being inspired by nature. We can design slippery surfaces that are effective in reducing marine fouling and reducing drag, and lots more. And that sometimes this inspiration might lead you down unexpected routes and leading to studying mechanisms that you had never even thought of before. My last slide is the acknowledgement slide. So the people that did all the work, mostly Chris, Sam, and Li Wen, and some of the other people's work I wasn't able to show, but I'd be happy to take questions and talk about this a lot more. So over back to you.
Chiara, fabulous to hear about her work. Wow, what a journey we have been. And I was just so taken by it because when Chiara started putting the pointer, which was a black thing, I thought, whoa, there's a fly near the pitcher plant. So really, thank you very much, Adam and Chiara, for taking us on this surprising journey. For Drew and me, when we were starting to talk with them, first 5 minutes, we learned so many things that we were surprised, like, oh, wow, man, that's amazing. So, so thank you. Now, I would like to invite questions from the audience for both our speakers. Can you please submit your questions by scanning the QR code on your screen or in the program? And I'll have a look as the questions come in. It's saying active questions 4, but I can't see any questions yet.
Well, in that case, maybe I'll jump in and kick off. I can see the questions. So here's a one for Chiara. You spoke about the radius of curvature being important. Is there too small a radius for these nanowrinkles, below which your slippery surfaces won't work?
Oh, very, very interesting question. Thank you. So the answer would be no, we have not found a system in which these liquids will not penetrate. But if you had a liquid that was less wetting, then that would totally be a problem, right? So if you had a material that for which the contact angle, the way that the liquid spreads on the surface, let's say the liquid didn't spread readily on the surface, then having a small diameter would completely stop the penetration, and therefore you wouldn't get an infused surface. So an example of that might be, rather than having the glass capillary that I showed with water, if you try and infuse that same glass capillary with mercury, you wouldn't have spontaneous uptake. In fact, the liquid will be pushed away from the capillary. So as long as you can optimize the wettability of the surface to your lubricant, you will get infusion probably down to, let's say, a diameter of a couple of nanometers. Beyond that, you'll get to the stage where you'll get molecular or atomic, let's say molecular level coatings on each surface. But in that case, you wouldn't describe that anymore maybe as a liquid. Although that's an aspect that we're really interested in. What is the thinnest layer grafted or attached on the surface that you can still call liquid? So we're doing research on that as well.
Drew, did you want to take the next question?
By all means.
Sure.
So now changing tact a little bit, and this is a question for Adam. So can you shed some light on why plants have become carnivorous? Is it something to do with the poor nutrient supply in their environments?
Absolutely, and that's actually one of the key reasons why Australia is so replete with carnivorous plants. Carnivory is one of many nutrient acquisition strategies that plants employ. So some species might produce proteoid roots or have associations with mycorrhizal fungi, or they may produce dorsiform roots or other strategies. It's all aimed at trying to ensure that they have better access to key macronutrients, primarily nitrogen, phosphorus, and potassium. And carnivory is the most effective when it promotes competition, or when it allows plants that are utilizing it as a strategy to be competitive against other plants. So for example, the characteristic carnivorous environment of being abundant, where you have high sunlight, very moist soil, very nutrient poor soil, and very wet soil, allows carnivorous plants to circumvent the low nutrient situation that they find themselves in, but also thrive in the high light and the high availability of moisture. So where we have very, very nutrient poor soils, so for example, here in Western Australia, we have some of the most nitrogen and phosphorus impoverished soils anywhere in the world. South Africa is another good example of that. We see very, very high levels of diversification in carnivorous plants because they have adapted to fill various niches within those ecosystems.
Thank you, Adam. Drew, I'll take the next question.
Sure.
So this is one which is for Adam again. Um, do you know if there is a decrease in arbuscular, whoa, that's a hard one for a physicist, mycorrhizae?
Arbuscular mycorrhizae.
Yeah, there you go. That's better said. Relationships in carnivorous plants. It would make sense if they have reduced need for nitrogen in particular due to their diet. Adam.
Yeah, that's a really interesting question. There are very few, if any, carnivorous plants where there is proof of an arbuscular mycorrhizal association. And in fact, some groups, for example, the Drosera, the sundews, the sticky-leafed species here in Australia and elsewhere, but Centre of Diversity here in Australia, so we claim them, they actually exude a compound from the roots called droserone, which is an antifungal agent which actively suppresses, or we suspect, actively suppresses the colonization of root tissues by mycorrhizae. So there likely is some association. For example, the Albany pitcher plant, there is a suspicion that there may be arbuscular mycorrhizal association, but it's either unstudied or unproven, and some species actively avoid it as a strategy because they've invested so much of their physiological energy into other ways of obtaining or mining for nitrogen, phosphorus, and other elements.
Okay, Drew.
So a question here for you, Chiara. So these non-slip surfaces, could they hold applications in areas such as machinery, where you could eliminate the need for traditional hydrocarbon lubricants?
Yeah, I guess the effect that we're after is a lubrification, very similar to what's traditionally done in friction reduction. I guess while the friction reduction aspect had been investigated, for a long time in engineering, the idea that you could have the same lubrification on a liquid interface, right? That's more recent. And once you know about it, it all makes sense. There's really a lot of similarity between the friction of between a solid and a liquid, and between, say, a solid and a solid. So a lot of those solutions, indeed, were already proposed for friction reduction in solid-solid contacts. I guess from an environmental point of view, it would be better to be able to do without silicone oils, as well as the other alkanes and other type of perfluorinated lubricants that are used. And we're also searching for materials that will be more renewable and that will have a shorter lifetime when released in the environment. But as yet, we don't have anything that's quite as good as the silicone oils.
The next question is again for Chiara. Do you know if the quintessential slippery banana has small wrinkles when observed by SEM?
Good question. No, I can't say I've looked at the peel of a banana, but I guess it's a very similar mechanism. Yeah, I think I absolutely get the picture that there is a porous, sort of soft, elastic structure in which the mucilage of the banana, that's quite liquidy, is sort of infused. And when you happen to try and step on that, that's definitely friction reducing. Other examples that people might have encountered of this type of mechanism is a lot of the steel pans that are sort of used in woks and things like that. You might be familiar with the idea that you have to treat them with oils. So you have to basically infuse them with oils before you use them. And then after you've washed them, well, really you should wash them only with water so that you don't remove those oils. And then you lubricate them again with the oil. And I think they're basically a lubricant infused surface. So they're non-stick pans because they use that oil within the structure of the steel.
Adam, Drew, is there any further question there?
Absolutely. I think we've got one here from the audience for Adam, and certainly this is a question that I'm interested in, is what differentiates carnivorous plants from carnivorous animals from a classification standpoint?
That's a really interesting kind of philosophical question. And I've seen a little bit of debate about this in the literature. Depending on how you look at it, and people have used terms like predatory plants to describe carnivorous plants. I mean, essentially, the only difference is that animals are motile. They're capable of moving. You know, things might be very different if a Venus flytrap had the capability to move around the landscape in search of its prey. But we have drawn parallels between carnivorous plants and carnivorous animals. So to illustrate that, there's a process that a lot of insects undergo called autotomy, which is where when they are attacked or frightened, or I don't know if insects get frightened, but under certain situations, they will actively lose a limb or part of a tail or something like that as a defense mechanism. And we see that that is a common strategy that a lot of different insects employ to escape, say, you know, birds or other mammal predators. Insects employ exactly the same strategy to escape from entrapment by carnivorous plants. We see plenty of examples of autotomized limbs and wings and things like that on adhesive traps like, you know, leaves of Drosera and often in other carnivorous plants as well. So I think the boundary between, you know, an organism being carnivorous is very gray, whether it's a plant or an animal. They are just as capable of undertaking targeted selective predation on organisms. It's just that they can't move.
Could you hear me, Chiara?
Ah.
Okay. What got you into studying biology for applications in physical chemistry?
Sorry, I missed the start. Was that for me?
Yes.
Okay. Well, I should reveal I know very little about biology. I can't say I've studied it in any detail, really. But you'll find that in the physical science, in the material science, often people go really fishing in the biological literature to find examples of animals or plants that have some important feature. Because, as I said, that is a very simple way to shortcut the research process. So some of the examples that we work on, for example, is the lotus leaf, which is a super hydrophobic, extremely water repellent, also slippery. And the reason is because it traps within the structure of the leaf air pockets. So not dissimilar really in the end to the pitcher plant, but in a different scenario. There's examples of animals. So the Namib desert beetle that has a structured back that is ideally designed to condense vapor from the atmosphere, and so enables it to drink water that condenses from the atmosphere. And whenever these phenomena are revealed in biology, then the physical chemist and the physicist and the engineer really pounce on it to try and use that to some benefit.
Thank you. Drew.
And so a quick question for Adam. Are they still discovering new carnivorous plants?
Absolutely. We are still discovering new carnivorous plants almost monthly. We've discovered new carnivorous plants here in Western Australia this year, even last year. There's been several published, you know, almost every year for the last decade and beyond. There was a new species discovered in Brazil by chance because someone had posted a photo on Facebook, and someone who knew what they were looking at saw that, realized it was different, and then went to the location and discovered that it was a new species. A large part of the issue, particularly here in Western Australia, is trying to ensure that there is adequate understanding and engagement with Indigenous groups who may have known about these species for literally thousands of years before Western scientists came along and began, you know, describing them in the way that we describe plants and animals. So I think that one of the key things in that discovery process now is trying to ensure that the sort of westernized science approach to taxonomy and systematics is now acknowledging that some of these have had cultural uses, or at least been culturally significant or well known to cultural groups for thousands of years.
Fantastic, thanks.
Can I have one more question? Okay, this one is for Chiara. So Chiara, you are able to hear me? Yeah. So is the viscosity of, so first of all, complimenting about your talk, but is there a viscosity of the surface liquid something which is important? Have you tried different coating liquids for different applications? Is there one that has been tried for coating surfaces which move in gas rather than liquids? For example, coating the surfaces of cars or aeroplanes.
Yeah, so we've tried a few different viscosities. So silicone oils you can pretty much buy in a very large range of viscosities. In the end, you find that there's a sweet spot where it doesn't take forever to infuse the surface. So, you know, students want to go home and get onto something else. So they want to get an infusion that will take, you know, say a few hours. And then on the other hand, you don't want something that's too, let's say, too liquid or too low viscosity, because that's been shown to deplete more rapidly. So somewhere around the 10 centistokes is what we're operating with. We often test 100 centistokes, and we haven't really seen an improvement in the longevity of the surface or the performance. One thing that might be interesting to play with, which we haven't yet looked at, is to play with viscoelastic liquids, so that they might have a particular shear at which they start. So yeah, the particular level of, say, pressure that needs to be applied to them before they start flowing. That might give you also very interesting phenomena. I haven't really looked much at the interaction of surfaces with air, so that aerodynamics, but a lot of the structures that we've studied that reduce drag in liquids will also work in air. And so, for example, there's things called riblets that people can put on airplane hulls to reduce friction in that setting. So similar rules apply there.
Okay, so I'm being signaled that we should bring it to an end. There is a huge lot more questions. Typically, if you were here, there'll be people queuing up to ask those questions. But unfortunately, we are on online mode. And so thank you very much for the great questions. There are more here, but I'm afraid that's all we have time for this evening. Chiara, one last question, because I'm standing here and I have the microphone. Can I send my glasses to you to coat so that raindrops don't come onto me? So, and I can hear the laughter, so many people here would love that as well. So, okay, a big thank you to both Adam and Chiara for joining us tonight, and to my co-convener, Drew, and you, the audience, of course. And we would like to thank Gira at Gia Station Wines and Hauberg catering for refreshments earlier this evening for those of us who were fortunate to be here in person in the Shine Dome. Thank you all, those here and those joining us online, for being with us tonight. And we do hope to see you again for the next installment of Surprising Science on 12th April. So thank you very much, and good night, everyone.